Thereafter, Adriana was a postdoc at Montreal Institute for Learning Algorithms, advised by Joshua Benjo, where she investigated deep learning techniques to tackle biomedical data challenges. And now Adriana is a research assistant at Facebook AI Research and a young professor at McGill University. So Adriana, the floor is yours.
I don't need it. Okay. So thanks for the introduction, and thanks for inviting me today to give a talk on deep learning for genomics and graph structured data. So before I start digging into the genomics part and graph structured data, I would like to go over, once again, over the deep learning promise, which I'm sure most of you have already seen that many, many times, but I still think that it's worth reviewing before motivating the work that I'll present afterwards. So we have all seen an increasing number of papers and successful applications of deep learning, and this has mainly been due to three key factors. So first, the increase of large-scale data sets, such as ImageNet in computer vision, for example. We have also experienced like a huge increase in computational powers through GPUs, and also the development and enhancement of deep learning models. So I will concentrate the talk on this last part, on the deep learning models. And we have seen successful application of these the deep learning models in many fields, such as computer vision, natural language processing, speech, and so on. And these deep learning models are currently reaching other fields, such as the healthcare field, with the hope that they will be at least as well performing as they have been in other fields. So before I move on, I would like to discuss a bit what is special about these models. So I cannot talk about models without talking about applications, because most of the models, we have to adapt them or extend them to tackle particular applications. So I would say deep learning was mainly popularized because of AlexNet back in 2012, when it won the ImageNet competition by a large margin with respect to more traditional computer vision approaches. And since then, convolutional neural networks have been widely applied to tackle other problems. For example, they have been endowed with region proposals to tackle object detection, or with an upsampling path to tackle semantic segmentation. And something similar has happened for sequential data, where RNNs are usually the model of choice, or RNNs coupled with attention have been very popular for machine translation tasks. And in between these two worlds of vision and sequences, there are other kinds of problems that usually combining convolutional neural networks and recurrent neural networks seems to be a good approach. However, there are other fields, and this is the case for personalized medicine, that involve a huge diversity of data, and where it's still not clear, or we don't have very well-established models. And in particular, in this field, I will concentrate the next part of the talk in two topics. So I will devote the first part of the talk to genomics, and what genomics data, what kind of challenges genomics data poses to deep learning models. I will go over a model that we proposed last year, present the data set where we tested our model on and some results, and then the second part of the talk, I will switch gears and talk about a completely different topic, and graph structured data. So hopefully I will convince you that graphs are an interesting structure to deal with. I will motivate why these graphs might be relevant to this community, and I will go over some of the prior work to motivate our contribution. I will present the work that we presented a couple of months ago, show you some results and potential applications that will hopefully be relevant for you. So let's start with the genomics part. So this work was a very collaborative work with many researchers and students at Montreal Institute for Learning Algorithms and at Institut Cardiologie de Montréal. And it was presented last year at iClear under the name of Diet Networks. So let me introduce you to the kind of data that we were dealing with. So modern genotyping techniques usually target millions of variants in the genome, and that's what we call the single nucleotide polymorphisms, also known as SNPs. So we will have a data set that is composed of such SNPs. However, even for large-scale studies, the number of participants that we have is still limited, and what happens is that we end up with this kind of data sets where we have a number of samples or number of participants, which is much smaller than the number of features that we have for each one of these participants. So we have a dire imbalance between the number of samples and the number of features, and this poses certain challenges to deep learning models. So First of all, what happens is that given the high dimensionality of the features for each one of the samples that we have, we end up having a parameter explosion, which is usually in the first layer of the network, which is the one where we have the number of parameters which grows linearly with the number of input features. So in a naive setup for SNP data, we can think of having a number of inputs that is around the hundreds of thousands, while having a number of samples that is around the order of magnitude of thousands. So given this setting and this parameter explosion, a natural question that arises is why don't we use convolutions? So in this case, we would have parameter sharing, and that would mitigate a lot this parameter explosion. However, when we're talking about this SNP data, we're extracting SNPs that might be very close together in the genome or very far away from each other. So we don't have this locality to exploit. And convolutions are good to exploit spatial locality, and they are suitable for data that has a grid topology. However, in our case, we do not have access to the full DNA sequence, and thus other kinds of models seem to be more appropriate. Given this parameter explosion that I was discussing, we have more chances to fall into the overfitting regime. And moreover, because of this high dimensionality of the data and the few number of samples that we have, we end up having very sparse data points in the input space. So what we propose is what the framework that we called dyad networks is a neural network reparameterization, which has as aim considerably to reduce the number of free parameters that the model has. And it's specifically designed for the setting where we have the input dimensionality, so the number of features in our data set being much orders of magnitude higher than the number of samples that we have. So let's dig into the model in more detail. So again, remember the setting. We have an input data matrix, which is of size n times f, where n is the number of samples, and it's much smaller than the number of features f. So if we think of a standard predictor network, a fully connected predictor network, we would have as input, in this case, one sample of dimensionality f. This sample, we would feed it to some set of fully connected layers, and eventually we will end up with a prediction. Additionally, in this kind of models, we can also add a second leg that would perform a reconstruction of the input. So in this kind of models, the heavy layers are the first layer of the model, because it's the one connected to all the high-dimensional inputs that we have. So it's the one where we have the exponential growth, the linear growth with the number of features of parameters. And the same happens with the reconstruction layer, since we're trying to reconstruct all these features, all these high-dimensional features that we have. So our goal here in this model is to reduce the number of free parameters of those layers. And to do that, what we did was to build a second network, which we called an auxiliary network. And this second network, instead of taking an input sample from the data set, it took all the features of the data set. So it's like we have the data set, and we transpose the matrix, and we start using the samples, taking into account the features instead of the individuals. So we feed this network one feature, so that would be all the values for one particular SNP across the training data set. And we feed it to a network, eventually going through an embedding layer, which could be a learned embedding or a handcrafted embedding, adding some extra transformations and nonlinearities. And we would use these embeddings that we learned as parameters of the initial network. So the idea behind that is that if we have a feature, we're learning a feature embedding. And we would like that two SNPs or two features that are similar or close to each other under some circumstances will end up having the same similar embeddings. And we will use these embeddings as weights, so they will end up having similar weights in the projection of the predictive network. So now, how is this reducing the number of free parameters of the network? So if we think about the initial network, and we take the example where we have 300K input features, and we want to reduce the dimensionality into 100 features, if we only take into account this network and we allow these parameters to be free, we end up having about 30 million parameters only in this first layer of the network. However, if instead of having these parameters we tie them to the second network, because the second network will be predicting them. Then what happens is that, assuming that we have an embedding of 500 and a single layer of 100, in this case, we would have only 50K free parameters. And to further convince you that this matches in terms of shapes, what happens in the first layer of the network is that we have a weight matrix that has dimensions f times 100, so f being the number of features, 100 following the previous example. And the output of the auxiliary network is a 100-dimensional vector for each one of the features in our data set, so that matches. And we can apply the same reparametrization to the reconstruction of the input if we have this leg. We can share parameters between those two networks up to any layer we want. So now that we know how this diet network frameworks looks like, we've seen that there is this embedding box in the auxiliary network. And so we played a bit with different embeddings, and I will go over two of them. So one of them would be the simplest embedding, so that would be just an extra layer in the auxiliary network that we would learn end-to-end with the whole system. Or what we call the per-class histogram, which is a handcrafted embedding that is often used in the genomics community. So I will give you a bit more details on what a per-class embedding is. So let's assume we have this data set. So I transpose the metrics for convenience. So we take one of the features, so one of the SNPs, and we get different values, which in our case can be either 0, 1, or 2 across the population. I put two different colors to simulate two different classes. So we have red class and blue class in this case for each one of the individuals in the training set. So we take class 1 and we count how many zeros, how many ones, how many twos we have for this particular SNP, and we have some frequencies for each one of those values. And we do the same for the second class and for as many classes as we have in our problem. Once we have that, we can concatenate all this information, and this gives us this per class histogram embedding that we can use in the whole pipeline. So now that I covered the diet networks framework and some of the embeddings that we used, let me show you some of the results. But before, I will need to introduce you to the data set that we used, which was the 1,000 Genomes project. So for those who are not familiar with the 1,000 Genomes project, the 1,000 Genomes project is a large-scale comparison of DNA sequences. And this comparison is made thanks to the presence of genetic variations. So this means that we do have access to SNP data for each one of the participants in the data set. The data set represents 26 populations, so we will be trying to predict ethnicities in this case. And these 26 populations belong to roughly 5 larger geographical regions. In our case, we used about 300K SNPs, and these SNPs are encoded as having either 0, 1, or 2 copies of a genetic mutation. So here are some results. So the farmost right result is just the predictor network without having the reparametrization. The x-axis represents the number of free parameters of the networks in the log scale, and the y-axis represents the misclassification error in percentage. So we can see here that we have the initial predictor network, the fully connected layer, the fully connected network, and the diet networks approach, and we see that we have a significant drop in number of free parameters, and we get better results. We compared that to two simple PCA approaches that they use in the genomics community. It's just a PCA with some classifier on top of it. We took 50 as number of principal components because that's the number that they would usually use for this kind of data set, but also tried with 200 because that was the one that gave the best results. So given these results, we showed the potential of deep learning techniques also to tackle genomic-related tasks. So let me show you a bit more of the results. So those are the confusion matrices for both the 26 populations and the 5 larger geographical areas. So this was the results of a network that was trained on the 26 populations and not on the levels of the 5 larger geographical areas. So as for the populations, we see that the network makes some mistakes, such as confusing different populations of Indians, or confusing residents of Utah with northern western ancestry and some participants from the UK. If we instead consider the larger and broader geographical regions, we see that the prediction becomes easier. So something that we did after seeing the results, and also because our collaborators at the Institute of Cardiology in Montreal were interested in that, is try to understand what the network is learning. And to do so, we did some t-SNE plots of the input of one layer that is the first layer of the network and the layer that is closer to the output. So let's see how this looks like. So that's the t-SNE for the raw input. So we see that all the populations are kind of overlapping. After the first layer, if we see that small clusters start appearing, and roughly what happens is that each one of these clusters roughly corresponds to each one of these 26 populations. And if we go higher in the hierarchy of layers, what happens is that as we get closer to the output, these small clusters start gathering together. So we did the same visualizations, but instead of taking into account the 26 initial populations, we took into account the 5 larger geographical regions. And what happens is the following. So we see that in the second layer, what happened is that this gathering together to build larger clusters are roughly correspond to the 5 clusters corresponding to the 5 larger geographical areas. So to wrap up on the genomics part, we introduced the diet network, which is a model reparameterization, which allows us to reduce the number of free parameters of the model and is specifically designed for cases where we have very high dimensional inputs and where we have a dire imbalance between the number of features and the number of samples. And we showed the potential of deep learning techniques to tackle genomic-specific tasks. OK, so now I will move on and switch gears to a completely different topic. So this topic is related to graph structured data, and that's also a collaborative project between researchers and students at Mila, University of Cambridge Computer Vision Center. And this project was mainly led by Petar Velikovic, who was an intern at Mila last summer. And this work was presented at iClear a couple of months ago. So the motivation is that I would say graphs are everywhere, and also in the biomedical domain. So we can think of molecular fingerprints. We can think of protein-to-protein interaction networks. We can think of gene interaction networks. We can think of epidemiology networks. And we can also think as brain connectons, as we have seen yesterday in the oral presentation of Guillaume Cucuruy. So I hope this is a strong enough argument to show you that graphs are present in the biomedical domain. And before I dig into what we did in our work, let's go over the problem formulation. So the problem that we wanted to tackle was the node classification problem. So basically what we wanted to do is assign one label for each one of the nodes in the graph. As input, what we have is a matrix of node features and also an adjacency matrix that gives us the connectivity of the graph. As output, what we have, or what we expect, is a matrix of node class probabilities. In this talk, for simplicity, I will assume that the graphs that we'll be dealing with are undirected and unweighted. But for most of the models that I will discuss, they are readily applicable to a broader set of graphs. And I will focus on the inductive learning problem, which is a problem where at training time, we do not have access to all the nodes of the graph up front. And this means that these kind of problems are suitable for graphs where we have nodes that are appearing, or for graphs that are completely disjoint, and we want to use some of them for training, some of them for validation, some of them for tests. So basically what we want to do is generalize to unseeing graphs. So in order to motivate our contribution, I wanted to give you an overview of the literature in the neural networks for graphs. So this is by no means going to be a detailed overview on all the literature that we have, but it's just to give you a sense on what previous methods have tried to do and to motivate our contribution. So the simplest thing that we can think about in the case of graphs is to build a per node classifier. So a per node classifier basically would expect a set of features that would be the node features to be fed into, for example, a multi-layer perceptron in order to classify it. So in this case, we would be treating all the nodes in the graph independently. So the problem with this approach is that we would not be exploiting the graph structure at all. So there have been some attempts to overcome this and introduce some kind of constraint or some kind of additional structural features to the node classifier. So this is the case of the paper introduced by Weston et al. In 2008, where they introduce a node similarity constraint that basically tries to enforce that nodes that are connected try to be close to each other in terms of features, and nodes that are not connected by an edge force them to be farther away. And this is also the case of these other papers, which basically try to introduce or learn some structural features and concatenate them with the node features. And this model is reminiscent to the skipgram models of the natural language processing literature. And they define a good representation as a good representation as a representation that should have should allow us to predict the representations of the neighboring nodes. What happens with this kind of approaches is that we are introducing the graph structure, but we are introducing it in an indirect way. There are other models in the literature, such as graph neural networks or gated graph neural networks, which follow a propagation model, and they feature this propagation operation at different steps. So these kind of models are reminiscent to recurrent neural networks, and recurrent neural networks were specifically designed or seem to be well suited for kind of sequential data, and our graphs are not this kind of data. So in our case, we have static features, so it seemed reasonable to explore all the other kinds of approaches. So the question now was, how about leveraging convolutions? And I hope Guillaume already convinced you yesterday why convolutions could be helpful in this kind of settings. So just to remind you of some of the arguments. So first, convolutions are independent of the input size. They have a fixed number of parameters because we have this weight sharing scheme. They are localized, so they act on local neighborhoods, and they specify different weights to different nodes in the neighborhood. Moreover, they are readily applicable to inductive problems, which is what we care about in this presentation. The question then is, how do we apply convolutions on graphs? Since each node in the graph might have a different number of neighbors, and it's also not straightforward how to slide this convolutional kernel over the graph. And there have been many approaches in the literature trying to build graph convolutions. And there are two main research directions, so one involving spectral approaches and one involving non-spectral approaches. So I'll start with the spectral approaches first. So spectral approaches operate directly in the spectral domain. So this means that they build a graph Laplacian, which is defined as the degree matrix minus the adjacency matrix of the graph, or some normalized version of those. And they compute the eigendecomposition of the graph Laplacian. And this is what allows them to transform the graph signal into the spectral domain. And once we are in the spectral domain, convolution becomes a simple multiplication. So that's how a convolution would look like in this kind of network. So we see that the signal is transformed into the spectral domain, then the convolution is performed, and then it's transformed back to the graph domain. So some potential limitations of this kind of approaches are that, well, first we need to compute the eigendecomposition of the Laplacian matrix, and this can be expensive, especially for large graphs, so graphs where we can have a lot of nodes. And moreover, filters are not localized. So in order to try to overcome these limitations, there have been other literature like the CHEBYNETS or the graph convolutional networks, which rather than computing the Fourier transform of the graph, what they do is use a Chebyshev polynomial approximation of order k. So this is how it looks like. I didn't write into the equations of the Chebyshev polynomials, which here are the tk of L. But you'll have to believe me that these polynomials have a recursive formulation, which highly simplifies the computation. Moreover, this Tk is a weighted sum of all the powers of the Laplacian L. And when we take the powers of the Laplacian, what happens is that we are able to see more hops away. So we end up having an operator that is k localized. Moreover, in this case, we have the number of parameters which is fixed to k. So as we can see from the equation, there is one parameter for each one of the neighborhoods. And since we have one parameter that is the same parameter for all the neighborhoods, what happens is that we end up learning isotropic filters. So it means that we are unable to specify different weights to different nodes in a neighborhood. So we wanted to address this problem. And going to the other main research direction, which are the non-spectral approaches, I will only cover a couple of them. So one feature, one characteristic of these kind of approaches is that they do not assume any particular and fixed graph Laplacian up front. So they operate directly on the graph domain, and the question is how they handle the fact that each node in the graph might have a different number of neighbors. So there have been the molecular fingerprints networks where what they did is learn a different weight matrix for each node degree. And in that case, that was reasonable because for their particular problem, their node degrees varied from 2 to 5. But clearly, if we want to scale that to graphs where we have like a very wide degree distribution, this might not be the best option. There has also been the graph sage approaches. And in their case, in order to keep the computational fingerprint fixed, what they do is they restrict the degree of each node to be the same. And the way they restrict it is by sampling a fixed size neighborhood for each one of the nodes. But this, in this case, we are dropping some data that might end up being relevant. So our approach to these problems is the graph attention networks. And the idea behind the graph attention networks is to leverage the self-attention operator in order to emulate convolutions on graphs. So just a quick overview on what the self-attention operator is. So self-attention was introduced in the context of text, but we can easily apply it to graphs in the sense that the idea is to allow the input to attend over itself. So the way to describe it is by means of these two following equations. So we can think we have a node I in a graph and a node j, and each one of these nodes has some features, so hi and hj. We will fit the concatenation of those features to and transform them through a network, and that will give us a score. And once we have the score for all the pairs of nodes, we can apply a softmax classifier, and that will give us some attention coefficients, which we can use as weights of our convolutional kernel. So a naive formulation in this case would compute the attention coefficients among all the pairs of nodes in the graph. And this would drop the structural information. So in order to avoid this, what we do is that we restrict our model to only compute the attention coefficients over some particular neighborhood. So we are making our operator localized. So here is a bit more into details what we did. So this is the network that computes the attention coefficients. So we see that we fit to the network some linear transformation of the features of two nodes, two connected nodes in the graph. And we feed them to a network that will give us a score. And once we have all the scores for all the pairs of nodes in a neighborhood, we can compute the softmax, we can apply the softmax nonlinearity. So in this case, we have the E equation where we compute the scores, and then once we have the scores, we can apply the softmax and get alpha, which would be the attention coefficients. So once we have these attention coefficients, in order to emulate the convolutional operator, what we do is apply them to a transform version of the features of each node in the neighborhood, and we aggregate all the features in the neighborhood. And this is the result of the output of a convolution, and we can stack as many of these graph attention layers as we want in our network. And these are the results for one attention head. But if we want to further increase the capacity of the networks, we can compute as many attention heads as we want. So this means that we will repeat this process as many times. In the case of the figure here, we are computing 3 different attention heads. And in the end, we need to either concatenate them or average them in order to get the features, the output features of the layer. So now that you have an idea on graph convolutional networks and our contribution, I will present results on 2 data sets. So the first data set are the protein-to-protein interaction, is a protein-to-protein interaction data set. It's composed of 24 graphs, and each graph corresponds to 1 human tissue. These graphs can have different number of nodes and can have different connectivity. As features, each node can have positional gene sets, motif gene sets, or immunological signatures. And as output, what we expect, so this is a multi-label classification problem, so we would like to end up having n active labels where each label represents a protein role. And the second data set is the one that was introduced yesterday, coming from the Human Connectome project. In this case, we have 100 graphs or 100 meshes, which represent the brain of, the cortical part of the brain of each one of the subjects. In this case, all the meshes have the same number of nodes and the same connectivity pattern. The features that we use are some structural features and some functional features, and the output is to classify each one of those nodes as belonging to area 44, area 45, or none of those areas in the brain. So here are some results on the protein-to-protein interaction data set. So the results are presented in percentage of F1 score, and we see that the graph attention networks are the one in the farmost right. So we see that the graph attention networks are actually outperforming existing benchmarks on the literature. So I will not go over the results that were already presented yesterday, but just as a reminder, we see that in the case of cortical meshes, we see that both graph convolutional networks and graph attention networks are the ones doing a better job with respect to other methods in the state of the art. So I don't want to wrap up the graph convolutional part, or the graph attentional part, without also going over a late contribution of some of our collaborators on PARATOPE prediction. So in this case, what they want to do is to have some antibody amino acid attend over the antigen amino acids to predict the part where they bind. And here are some of their visual results. So in the middle picture, we have the antibody, and with the different colors represent the probability of binding with the antigen. And on the picture of the right, you have the attention coefficients for a given amino acid, which they know actually binds. So to wrap up with the graphs part of the talk, so we introduced the graph attention network, which is a model which implicitly allows us to assign different weights to different nodes in a neighborhood and operate directly in the graph domain. It provides localized operators. The network itself does not depend on the upfront access to the graph Laplacian of the graph structure, so this makes the approach readily applicable to inductive problems, which are like the 2 problems that I presented today. Moreover, it has a fixed number of parameters, and this number of parameters only depends on the number of features and does not depend on the node count or the degree of these nodes. And moreover, we can argue that it's computationally efficient from the point of view of self-attention being parallelizable across edges, but also the computation of the output features is also parallelizable across nodes. Finally, I presented also the 2 potential applications of these graph attention networks in the biomedical domain. So that is the protein-to-protein interaction networks and the cortical mesh parcellation application. So to wrap up this talk, I just want to remind that in order to move forward to personalized medicine, we need to take into account that for each one of the subjects or for each one of the patients, we have a huge variety of multimodal data. So this includes, but it's not limited to, text in the form of unstructured text that can come in electronic health records or examination results. We have a bunch of imaging modalities, MRI, CT scans, which we are very familiar with. We also have access to omics data, so genomics, transcriptomics, etc. And we can also, and hopefully I have also convinced you that there are a lot of biomedical data that can be represented as graphs. So that's the case of gene networks, gene interaction networks, protein interaction networks. But we can also think about these graphs as a way to connect the different information that we have, or different patients, or study other things such as how diseases spread, for example. And I cannot end this presentation without thanking all my collaborators, because this work wouldn't have been possible without all of them. So collaborators in Montreal Institute for Learning Algorithms, at Institute of Cardiology, the Montreal University of Cambridge, and the Montreal Neurological Institute. So thanks for your attention.
Thank you for the nice talk. Are there questions from the audience?
Hi, very interesting talk. For your first part, for the DIANet, I'm just curious if there's any way you can tell from the SNP embeddings which ones are predictive, which ones are not, just like the Manhattan plot people usually used in the GWAS cases.
So if I understand correctly, what you're asking about is the interpretability of the feature representations that are learned by the auxiliary network, so like the embeddings learned for each one of the features. So I haven't presented any results on that direction. That's something that some of our collaborators in the Institute of Cardiology de Montréal are looking at. So they wanted to see how this SNPs cluster in terms of different functions. But so far, we don't have any results to share.
Thanks for an interesting talk. So typically, when you have many more features than data points, you can go to a double space. And I know this is cursing in the church, but this is what kernel methods would do in the old days. I wonder, when you compare, did you compare to simple like SVMs with non-linear kernels, or maybe to kernel PCA instead of PCA?
No, we didn't do that. That's a fair point. We could do that, and that would maybe make the conclusions of the model stronger. So thanks for the hint.
Okay, if there are no more questions, I'd like to thank Adriana again.